रोहित जी, मेरी आवाज आ रही है?
यस मैम।
देखिए आप मिस्टर अजय को, सईदा को और रिवांगिनी जी को, इनको को होस्ट बनाइए। रिवांगिनी रांझा जी को भी को होस्ट बनाइए। रिवांगिनी, आप अपना कैमरा और माइक एक बार चेक कर लीजिए प्लीज।
हाँ, ओके मैम। एम आई विजिबल मैम?
यस यस, वेरी गुड।
ओके मैम।
वी शैल स्टार्ट बाय 6 PM।
ओके मैम।
ओके। Dr. Tanushree will be joining in a moment, maybe by 6:10. All the participants to stay attentive. Dr. Tanushree will be joining by 6:15. She has texted her now. Just wait for 5 minutes more. We'll start the session shortly. Rohit, Dr. Tanushree ma'am has joined. Please make her co-host. Good evening, ma'am.
Hello. Am I audible?
Good evening, ma'am.
Good evening, Pragati. Am I audible, right?
Yes, ma'am. You're audible, ma'am. How are you, ma'am?
I I had been down with fever yesterday and the whole
Oh,
night. So I was slightly off my balance, but I said, no, I promised and I have to keep my word. So that is why.
thank,
Yeah,
thank you
I
so much, ma'am.
welcome.
Ma'am, shall we start?
Yes.
Okay. Rivangini, please start the session.
Okay, ma'am. Am I audible?
Yes, Rivangini. Good to go.
Okay, ma'am. Good evening to one and all present here. I, Dr. Revangini Ranja, Director HR at AgriMeet Foundation Bharat, warmly welcome you all to 3 months International Agri Training cum Internship Program, Krishi Navyog. This program has been designed with the vision of connecting young minds with scientific innovations, advanced agriculture technologies, and inspiring personalities from field of agricultural research and biotechnology. Today's session is truly special because we are honored to have with us an eminent scientist, visionary researcher, and inspiring personality, Dr. Tanushri Kaul, Group Leader at the International Center for Genetic Engineering and Biotechnology. Dr. Kaul is internationally recognized for her pioneering contribution in the field of plant molecular biology, genome engineering, crop biofortification, plant nutrition, and stress biology. Through her remarkable research and scientific leadership, she has emerged as a leading authority in developing sustainable biotechnological solutions for global food and nutritional security. Ma'am completed her PhD in botany from the University of Delhi and has built an illustrious scientific career dedicated to crop improvement and advanced genome editing technologies. Her expertise include CRISPR-Cas9 technologies, plant stress physiology, nutrient enhancement, and molecular breeding approaches for developing climate resilient and nutritionally enriched crops. Her groundbreaking research focuses on combating micronutrient malnutrition and enhancing agriculture sustainability through genome engineering and biofortification. She has successfully developed phytase rich tomatoes, phytate free cereals and legumes, and wheat with enhanced micronutrient remobilization. Dr. Kaul has also worked extensively on advanced genome editing strategies in crops such as rice, maize, soybean, pigeon pea, and cotton to improve productivity and herbicide resistance. One of her most inspiring achievements is the development of genome edited iron and zinc rich rice aimed at reducing anemia, stunting, and hidden hunger among vulnerable populations. Her work beautifully connects crop improvement with human health, showcasing the transformative power of agriculture biotechnology in enhancing global nutritional challenges. Throughout her distinguished career, Dr. Kaul has received several prestigious fellowships and awards, including BioCare Award and Fellowship, Rapid Grant for Young Investigator Award, Indo-Israel DST Fellowship, and DBT IISc Postdoctoral Fellowship. She has also been nominated for the prestigious Viva Award for Distinguished Scientist 2025 for her exceptional contribution to crop improvement and biotechnology. Ma'am, your achievements and contributions are so vast and inspiring that they cannot truly be covered in a single introduction. Your journey continues to inspire thousands of students, researchers, and young scientists across the globe. On the behalf of entire team of Krishi Navyog and everyone present here, I extend a very warm and heartfelt welcome to you. We are truly honored by your gracious presence. Now, without taking much time, I would like to invite our esteemed speaker, Dr. Tanushri Kaul ma'am, to kindly enlighten us with her valuable words and inspiring session. Ma'am, the platform is all yours. Please.
Thank you, Revangini ji, for this introduction. And I've been thinking really it's been a too long introduction, though I have not achieved as much as I wanted to yet. So thank you again, Pragati ji, for inviting me for the student UG and PG. I guess there are UG undergraduates and postgraduates here, right? Am I right?
Yes. Yes, ma'am.
So I like to share my slides, and I would like to just. Are my slides visible?
No, ma'am. Rohit, please help, ma'am.
Loading.
I'm sharing the screen now. Is it visible?
Ma'am, it's still like, can you please reshare the screen again, ma'am? What?
Is it visible now? Is it-
No, ma'am.
Is
मैम
it
स्क्रीन इज देयर लेकिन लाइक आपके बैकग्राउंड में शो कर रही है ये स्क्रीन या दैट्स फाइन फाइन फाइन
अब ठीक है?
Yes, ma'am. Yes,
फाइन
ma'am.
फाइन
तो मैं अपना वीडियो बंद कर दिया है जिससे कि वो आप स्क्रीन देख पाओ.
डन मैम डन डन
So good evening, one and all, and I am here, Tanushri Kaul, and I have the privilege to lead the nutritional improvement of crops group at ICGEP as a group leader, and the the overview that I would be giving you today would I'll be walking you through the latest techniques that have been identified and utilized for improvement of crops employing genome editing, and how it came up, what is the background, how is it repurposed, and eventually some of the products that we have developed in the lab. This is a short introduction about the talk that I would be giving. So if there are any questions, they can be addressed in after I finish, and you can just note down your questions and then ask me in the end. Is it fine?
Yes, ma'am.
Okay. Okay. Thank you, Pragati. And there was a male voice, so I don't know who that was. I thank him also. Who is this guy who, the person who just helped me?
Ma'am, Rohit.
Thank you, Rohit. So here we go. As,
Hello.
yeah. Thank you, Rohit. Thank you. My screen is visible and I'm beginning, right? So the title of the talk today is Engineering the Future of Food, Indigenous CRISPR Innovations for Climate Resilient and Biofortified Crops. So as we all know that crops provide us with food, feed, and fuel for human life and other consumable resources, thereby contributing to the progress of human race and society. And the world, is predicted to hit 9.6 billion by 2050. And globally, the demand for crops will increase by 100 to 110% in the next 30 years. So in order to feed and nourish this burgeoning population in the face of climate change, decreased arable land, and water shortages, there is a pressing need for innovation in crop breeding technologies to increase agricultural productivity and accelerate sustainable agricultural development. So as we all know that India is the third largest exporter of groundwater in the form of rice and wheat. And there are enormous amounts of water that is invested for the growth of these crops. So when we are transporting or when we are exporting these crops, we are exporting our groundwater in turn. And we know that in the recent past, we have seen that several cities in India had been predicted to run out of groundwater. So this is affecting 100 million people across India, and human race, we all know, is riveted to evolving and transmutating beyond the fossil fuels as a strategy to adapt to man-made global climatic changes. We all know about the green hydrogen we are looking for, and we all know how we are shifting towards solar and eventually green energies. And yet another frequently unnoticed climate change malefactor is the animal agriculture, which we forget, which we let loose because we think that all the in climate change malefactor mainly is the fossil fuel, but it is not so. So the second largest unnoticed climate change malefactor is the animal agriculture, and besides that is the monoculture of rice crop, which leads to release of greenhouse gases. So the second largest is the animal agriculture, and it is a major contributor to human made greenhouse gases and after fossil fuels, and it is a prime cause for deforestation and water and air pollution and loss of biodiversity. So the second cause is the monoculture of the same kind of crops, mainly rice, wheat, corn, mainly these cereals. So growing them in and out, season after season and year after year also is a cause for erosion of the the nutrients from the topsoil, and also the reduction in the organic matter. So you see that soil matters us. Why? Because soil is failing across the world every 5 seconds. A soccer pitch of soil is eroded, And it's essential that by 2050, around 90% of Earth's soil could be degraded. That is what is being predicted by the scientists all over the world. So what does this mean for people and the planet? And what can we do to restore a healthy balance to the soil we need to survive? So in that case, we need to see that we, retain at least 5% of organic matter in the soil. Generally, that 5% organic matter is so crucial because it contains more organisms, than there are on the earth, because there are microbiome. You see the important microbes that are there that help to create degradation of complex, chemical substances like the nutrients, like all the nutrients are in the combined form, in a salt form, and these microbes degrade it into inorganic phosphates and inorganic nitrogen, inorganic nutrients that is utilized by the crop. So whatever we eat is basically fueled by whatever is there in the topsoil. That is 12 to 15 centimeters. That is the topmost one. So that is most important. It is crucial that they say that just 60 years of topsoil is left, and hence we need to take care of this organic matter. At least it should be 5%. You see, a large percentage of our planet's arable soil is used for family farming. So you see in Asia, how much it is, Europe, America, South America, North America, Central America, South America, Africa. So most of it is for family purpose. Whatever is grown is being eaten within the family or the community. And sustainable soil management could produce like up to 58% more food, because eventually the arable land is decreasing, and whatever is available for us to share across the world is very less. And that also is being directed towards intensive farming, especially monoculture of few cereals or crops. And this is damaging the very soil to grow healthy crops. As the soil quality decreases, so does the nutritional value of the crops. Hence, more fertilizers, we are putting more synthetic fertilizers, more pesticides, and that degrades or destroys the organic matter. The moisture is gone, and the microbes are gone because of it. That is why our Prime Minister Modi ji is requesting us to reduce the utilization of synthetic fertilizers, because eventually everything is based on the delivery of all these things into the soil, and that eventually leads to degradation of these important microbes. So what our Niti Aayog and which was Planning Commission initially proposes is diversification of diets, and this is crucial to protect the topsoil degradation and human health. So what we are basically, in addition to improving the existing crops around us, we are also trying to, create a pathway or inception for diverse, underutilized, nutrient-rich crops into the mainstream markets, so that the consumers have a choice to, choose the right kind of food that is required, and we just do not blindly do monoculture. So there are different crops that we are working on, and these are millets amongst some of them. So millets are there, rice bean is there as a legume, important in nutritious crop, moringa is there, buckwheat. So all these are there in pipeline, but as of now, this is our plan that we need to target all these, including millets. So regenerative agriculture, such as new breeding technologies that is combined with CRISPR-Cas breeding. CRISPR-Cas, along with the breeding, means you are introducing new traits through CRISPR-Cas and then using those lines for breeding purposes. And also, along with crop rotation and growing complementary crops in the same field, can eventually expedite or facilitate maintaining soil quality, and even recover or revamp the degraded soils to good health. So this is one of the ways to boost cultivation of protein and micronutrient-rich underutilized crops in the place of monoculture, which would tackle micronutrient malnutrition. As we all know, that nature has been editing, altering, and rewriting genomes for a very long time. And we know that it has been millennia since this has been happening through evolution, through natural evolution and natural selection, enabling organisms, both animals and plants, with genomic variants to survive. Moreover, the humans have been employing artificial selection for domestication. For example, for the past 10,000 years, the quantum of crops that we are consuming have been domesticated from their wild progenitors in the last 12,000 years, as you all must have read in your UG or post graduation. So, for instance, this wild ancestor, has given rise to the modern day hybrids of corn that we see. So, this teosinte, so the hybrid corn that we see now is produced by the wild ancestor through domestication of crops. So this is what naturally evolution has changed the morphology, how it looks, how it tastes, right? This is not through human intervention. It is through this is through artificial selection, human intervention, when humans have been trying to utilize specific traits in crop breeding, and then eventually you can see that there are several methods for artificial selections used in modern day agriculture. Some of them you are aware of, like crossbreeding, that takes around around 10 or more than 10 years, mutation breeding, and then transgenic breeding, which has not been flourished in India because we do not like to propose or propound the eating of transgenics or GMOs. That is when you have an external exogenous gene in that crop, people do not want to eat it. They feel that they will become something abnormal, or maybe they will be increased in cancer or whatever. There are so many notions there, but eventually these are not scientifically backed up. But people like several people are against them. However, these have been successfully grown in USA, Indonesia, Vietnam, China, and all the growing economies. And the latest one is the genome editing approach, which is the fourth generation technology that has been introduced. And amongst these, crossbreeding and mutation breeding are labor intensive, as you can see written here, and that they lead to genetic erosion and or reduction in the genetic diversity, because we are focusing on a few traits and few crops. Hence, we have recently seen that there is a narrow genetic diversity of cultivated crops. And this is a prime cause for yield plateaus observed in the breeding programs. And according to a recent report, we have seen that there in 120 essential rice genes, 120 essential rice genes have, that are governing the quality and the yield traits. They revealed just 28 of them, they were there, and they had a single haplotype. So you can read about what a haplotype is, and then you can see that if there is only a single haplotype for in a 3000 K panel, how much important is genetic diversity, and how much we are, reducing or degrading it through just focusing on few crops and few traits. You read about that. And hence genome editing, you see, is a tool that can reconstruct this genetic diversity in beneficial alleles or genes, and thus presenting the fourth generation breeding technology, which has emerged as a precision design agriculture via integration of deep exploration on crop functional genomics. Different kinds of studies have been taken up, molecular studies at the molecular level, at the physiological level, at the omics level. So these are all combined along with popular precision editing technologies and big data mining that are synergized into breeding technologies, and thus they can reconstruct genetic diversity in beneficial alleles. So if it can be employed to accelerate domestication of wild species or distantly related species like orphan crops by multiplexing of orthologs by main domestication genes. So this is like redomestication. For example, redomestication of a potato, which is an autotetraploid, by CRISPR has been done, and it has been converted into a self-compatible diploid line by just knocking out a specific gene. It's a SRNase specific gene, which, and then there are certain other aspects also, like there are vegetatively grown crops like saffron and banana and all these. These can also be, there are no germplasms for them, so these can be, their domestication can be accelerated through genome editing, which with a new technology that does not require the gene but just the protein, like the ribonucleoprotein complex is required, where the Cas9 protein does the work. We do not require the gene integration. So these are, so this is the technology. In 2020 it came up, and the people, they got the Nobel Prize in chemistry. But eventually the main aspect is that the humankind is now at the verge of genetic revolution, because it's not touching just crops, it is touching everything that's possible. We recently must have heard about the sickle cell anemia can be, treated through CRISPR based technology, right? The Cas9 technology. And then there are other aspects where you can take up human health aspects like infectious diseases, cancer immunotherapy, diagnostic tools. During COVID, you would have seen several tools that have been utilized. Then livestock animal models have been developed to knockout. You could use them for checking your drugs, drug delivery, and seeing how the drug works and all. So every environment, agriculture, plastics, biofuels, everything. So ready to use set of targeted surgical tools that have been poised to protect or recover our food supply from all the climate change or the weeds, poor nutrient levels in the soils, and pathogens. So this is the future breeding of crops through de novo domestication. So de novo, as you know, that there is no prior precedent of it. So something you say is de novo, that was never before there. So there are no novel traits available. For example, I'll explain to you. There, de novo domestications are promising, pathway for creating new future crops. So this is like, suppose if you talk to people, scientists who are working on increasing oil, production in mustard. So they say that there are naturally no natural variants or alleles present in the germplasm that can be utilized for breeding crops or mustard crops with higher oil yield. So they this is a place where these genome editing or genome editors like us can step in, and we can introduce a novel allele or a trait through de novo genome editing by studying the basic pathways and then seeing where the bottleneck is, what are the negative regulating genes or QTLs that can be identified, which can be knocked out, and not just by single genes, because single gene knockout or single gene modification may not work well in the fields, as we know, we have seen the precedents in the last few months. So eventually we need to multiplex more genes, target more genes which are from different pathways, so that we can multiplex them together and edit them together and then see the effect. Because most of the traits like yield and what you say, stress, they are all polygenic traits. So by just changing or modifying or knocking out one gene may not effectuate a robust trait in the field, in the farmer's field. So this is all. So this is what we are. See the genetic diversity here. It was initially so high, and eventually what we have done is, through domestication, wild plants that were grown during the early civilization time, and then the land races were developed through domestication. That took thousands of years, and eventually the breeders came up, and then they started using these land races to refine them into specific cultivars. Cultivars are cultivated varieties that have been grown in the farmer's field. So they are, they have limited selection, and now we are doing monoculture of these limited cultivars, right? So that is leading to a degradation of soil quality and the nutrient deficiencies in the soil. So what we propose is that de novo domestication is not just genome editing. It is starting with the identification of elite germplasm, creating if there is no mini core, or we need to develop a mini core, create a omics platform where genomics is there. You annotate the genes, you see their expression, then you see their protein profiles, and then you combine and create a network for each crop, which is a combination of all these aspects. And then you utilize your softwares to identify what targets, or use AI and machine learning based algorithms to target genes that can be effectuating basic traits that are like stress resilience, abiotic stress resilience, or nutrient enhancements. And that can be then done through genome editing, when you identify those targets and you predict what would happen before you edit. So that is more important. So that is where AI and generalized AI and machine learning based algorithmic designing help us. They are tools to help us to identify which targets, and how, if we do that, if we go about knocking out or introducing overexpression through promoter editing or what all. So that can effectually help us to eventually then go about focusing on that genome editing, rather than just randomly editing genes here and there. So then you can be grown. Then those traits, those events that you develop then can be given to the breeder, and the breeder can then use it for creating cultivars. And then we can do field evaluation, multi-location evaluation. So in that ways, your domestication, your genetic diversity is not lost. So this is what people have tried to use CRISPR systems for crop improvement, introducing complex traits into breeding strategies. For example, you know that there is a inverse relationship between the number of fruits and the fruit size in tomatoes. So you must have read about it. There's a paper on that by a specific scientists, when negative epistasis came about, and the two genes, initially the first gene gave more flowers and the second gene gave a bigger size, but they were inversely, they were they were negatively epistatic. So both together could not give a enhanced number of flowers and fruits and a bigger size. So that could have been that trade-off was there between these two traits. So then they use these specific genes to target them through genome editing to increase the fruit size, the fruit number, and the lycopene content simultaneously through multiplexing the CRISPR targets in one go. And then improving sometimes improving the promoter region, sometimes improving the upstream ORFs, whatever possible. So these are all different genes that have been targeted for domestication of tomato. And then naturally rice lacks in vivo haploid induction. So through genome editing, they've tried to create haploid induction that could eventually reduce the time taken to develop a variety. Generally it takes long years. So if you have a double haploid with a good trait, you just reduce those years to develop a cultivar. And then there are so many haploids that have been introduced by double haploids creating through multiplexing of these different genes that are involved for chromosome segregation genes. If you stop the segregation, then haploidy it can be leveraged. And then there are certain genes where that superior traits and F1 hybrids are lost. Every generation you have to create F1 hybrids. That's why companies sell their seeds every year to the farmers. So in this way, people have tried to streamline by multiplexing several genes to create in heterosis. And this heterozygosity of F1 hybrids was fixed by multiplexing of mitosis in place of meiosis genes. this is called MIME genes. So there are so many examples of different systems that have been generated with Rubisco, along with its, another gene that helps photosynthesis enhancement. So they've tried to create synthetic devices through synthetic biology, but utilizing genome editing tools to introduce modify industrial microbes, create green fuels, biomaterials for maybe space research, etc. So this CRISPR menagerie has, been so powerful and robust that it has now a huge control over the genetic composition of the species across the earth. So there you have, powdery mildew resistant barley and wheat. People have tried to increase the bacterial blight resistance in rice. People have tried to improve the, resilience of these sweet oranges that, were being, dying because of this disease, yellow dragon disease in California, Texas, Florida. Also, there are other soil fungi that was, creating certain diseases in this Cavendish banana, and people tried to create resilience to this fungus, and also many other aspects. There are certain gene drive experiments based on CRISPR that want to eliminate your female mosquito so that the transmission of malaria doesn't happen. So these are all aspects of cows with no horns. So even scientists are trying, like Beth Shapiro in University of California, Santa Cruz, that she's trying to study the relationships of the modern day species with the extinct species, that they're trying to bring them back live, which you had previously seen in movies. You remember the movie, I forgot, which tried to show that the dinosaurs have come back and they're there. So just to see what is the relationship, how did they evaporate away from the, so just to study what happened to them, and also these unique features they've been studying in different organisms. And we all know that it started from the point of the first, first in vitro tissue culture plant that was developed, right, and in 1960 the carrots, and eventually the new technologies came up, including the crossbreeding, mutational breeding, transgenic breeding, then marker assisted selections, and then more. So there are different other techniques that have come up, including genome editing, which have eventually led to the change. Recent emerging genome editing approaches led to the revolutionization of the plant breeding technology to levels, as such precision levels that never had been there before. And the first genome edited crop was the oleic acid, increased oleic acid content soybean, where because oleic acid is omega-9 and it helps in the increasing the temperature of the, the smoke point of the oil, soybean oil. So you can, if the oil at high temperature degrades, it leads to cardiovascular diseases, and rancidity leads to heart issues. So this omega-9 increased in this would lead to higher smoke points, so that oil will not degrade at higher temperatures when you're frying food or something. So I'm giving you such details, but I'll just skip some of these slides because I have just have to come to a certain point where you can just see what are the technologies. So these are the mutational breeding strategies that these two scientists got Nobel Prize for. And Mueller used the ionizing radiations to create artificial mutations. So these are all random mutations. So these are not targeted. So it can happen anywhere across the chromosome. So these are that drawback of random mutation is, lurking these aspects of chemical mutations, also ionizing radiations or this mustard gas. So what eventually was a first step for targeted genetic engineering was the gene targeting technology that was identified by Mario Capicchi, and he was a PhD at the University of a scientist at the University of Utah, and he got this Nobel Prize in physiology or medicine in 2007, along with two other components, scientists. And basically his question was that, because he created several knockout mouse models based on this targeting technique, which revolutionized the genetic and biomedical health and research, allowing scientists to create animal models for hundreds of human diseases. And his primary question was basically that if we can replace a perfectly good gene with a mutated one, right? So can we also go the other way around and replace the problematic genes with those that work? So that was his principle, and he utilized this gene editing targeting technology to enrich. It was a strategy to enrich embryonic stem cells that that were showing homologous recombination. It's like a screening process. You have a huge number of transformants, and you have to screen where actually the gene transformation has happened in the right place. So he utilized double selection strategy where there was a positive and a negative selection. So when they developed, so if it's like the homology donor repair technology that we utilize in our research. So he developed a small piece of DNA which had the intended mutations that he wanted to introduce in a specific site, in a locus in the chromosome, and he used HSV thymidine kinase selection and a neomycin selection. So when the piece with the desired mutation entered in the right place, the neomycin selection was used, the positive selection. They were positive for neomycin, but when it happened in the wrong place, then both the sides, when it happened randomly in the both selections, they showed positive. So that is how he selected these transformants. So you see here these two ladies empowered by their own intelligence and their what do you say, the power to envision the future and to see look ahead of time. So they were the ones who brought up this programmable dual RNA guided DNA endonuclease through studying the adaptive bacterial immunity, because they had a project. They had a project which was basically on how viruses attack bacteria. So this is how it was discovered, and they were the first to propose that CRISPR-Cas9 enzymes from bacterial origin had the power to control the microbial immunity. So these Cas9 endonucleases in the bacteria helped the bacteria to evade the viral attacks. So, so there was a programmable guide RNA, single guide RNA, which was 18 to 20 base pairs. Plus, there was a PAM sequence, a protospacer adjustment motif, which the Cas9 identified, and the target sequence guided it to create a double-strand break in the viral genome. So the bacteria survived. So this decision had to be taken within few seconds, otherwise the bacteria would die. So that is how they, repurpose this technology with by using the gene-specific 1820 base pairs as the target sequence, or you can say the spacer sequence, and the sequence that identified Cas9 was the protospacer adjustment motif, that was an NGG, and that without that NGG, the endonuclease Cas9 will not identify the target sequence. So that together worked as a complex. So this is how it originated. So there was an acquisition, and the viral DNA was inserted into the bacterial genome, and then there was a biogenesis process where the introduced DNA. Eventually, the whole array was created again through RNA transcription was done. So the RNA was produced, and then the sequence which matched with the so when the second time the virus attacked the bacteria, it would match the sequence and see if it is the same as that of the viral DNA, and then it would create a double strand break. So this is like a vaccination which we generally do. So this is how it evolved through the bacterial adaptive immunity, and now we have several several endonucleases. So among all the existing programmable site-specific CRISPR site-specific nucleases, CRISPR-Cas system has explicitly emerged as a prominently important tool and efficacious tool that facilitates the precise and targeted genome modifications in diverse organisms. You see, all these meganucleases, zinc finger nucleases, talons, they all came one by one. So first the meganucleases came up, then the zinc finger nucleases came up, then the talons came up. I don't have time to explain the difference here, but there are, these are all basically protein based, right? And this is DNA based, RNA based. So this is more easier to work and more cheaper and more user friendly. So you just need the 18 to 20 base pair sequence of the gene, and you can utilize that for creating double strand breaks in the DNA. And once the double strand break is introduced, the endogenous repair machinery inside the host or the plant or the crop or the animal introduced, identifies this break, and it tries to repair it. And when you try to repair that, it can be either done through the non-homologous end joining process, where insertions and deletions come up and they lead to gene disruption, gene function disruption, because there's a shift in the frame. And the other one is a homology directed repair process, which is precise DNA editing, and it requires a template with the desired mutation in the center, with two homologous left arm and right arm flanking this desired mutation. And when this is given, there is a site-specific homologous recombination event that happens, and there is introduction of this new desired mutations within the native host. So this is precise. It is error free, but this is not, this is like happening just less than 1% of the times, and most of the times NHEJ is happening. So you generally get knockouts easily than the homology repair. So amongst all these tools, programmable DNA cleavage endonuclease tools, you see the CRISPR-Cas system, which recognizes through base pairing between DNA sequence as a target site and then the CRISPR-based guide RNA. The Cas9 has two nuclease domains. So those two nuclease domains are shown here. So this is the RUVC and this is the HNH. So these are, or the HNH and the RUVC, these are shown by the red arrows, and this is how they cleave. This one cleaves this upper strand, this one cleaves the lower strand, and eventually the repair is done, which leads to introduction of the mutation. Now there are several genome editing tools here have been come up. There is Cas9 nuclease that came up, then there is Cas12a, 12b, 12c. So these are CPF, come under the CPF domain. Then there is Cas13 nuclease, which works only on RNA sequences. Basically, they are RNA-guided RNA endonucleases. Sometimes you require that, but then there are cytidine-based editors and adenine-based editors. So base editing also came up. So here this is here this is copy pasting and deletion and insertion. Here this is single base pair nucleotide changes can happen with this. And then primer editing. So I'm not going to do the details. So this is a overview of different kind of now Cas14 has also come up, and so there is a difference in their guide RNA sizes, and these are basically double strand DNA they require, Cas9 and CPF, and whereas 13 and 14 require, this one requires the RNA single strand, and this 14 requires a single strand. So there are a variety to choose from depending upon what work you want to do, what kind of cleavage you want, what kind of mutation you want to introduce. So as of now, the relevant plant genome editing tools have been type 2, that is Cas9, and type 5, which is CPF1, or Cas12 series A, B, C, D, E. And these have different domains and all, but eventually you see that here you see there is a staggered end that is produced through CPF1, and this creates a blunt end. And they said this is more, suitable for homology donor repair, whereas this one is more efficacious for non-homologous end joining. But these are simple and efficacious nature of they're simple and efficacious nature of engineering and adaptability for multiplexing, not just one gene. You can edit several genes together, helps in the efficiency in creating genome-edited crops. So this is how the efficiency is being shown in a cartoon in this GIF file. So you remove whatever is there and you introduce whatever is desired. See, so this is how, but the selection and all process that has, which gene, what what target you have to use, why you want to do it, what strategy you want to do, that you have to think and plan before you start the experiment. And you can read this paper of ours, and there are several others following this that can give you all the information here. But the main point is that whenever you go for whole genome editing of that crop, you need to have the whole genome sequence in your hand, because you need to develop your guide RNAs based on the software, and the software requires, or it blasts, uses blast software, and there you need to blast the entire the guide RNAs with the entire genome, and see whether there are no mismatches to avoid off targets. It has to be targeted, not off targeted. So we need to select. So that moment you need the whole genome sequence of the crop. So you need to have it. If there is a crop which has no genome sequence in the NCBI, you will not design the guide RNAs, and you have to first create the sequence. And also negative regulators of beneficial traits. All genes and QTLs that are negative regulators of beneficial traits are the simplest target for genome editing or for loss of function alleles. And single gene traits are easy to target. For example, quality trait like grain amylose content, or oil quantity like omega 9 if you want to increase. So only one gene is involved in that. And if you want to increase the amylose content, any single one gene is involved in it. So that helps in targeting and improving the quality. So but if there are quantitative traits like yield and stress, then you have to target multiple genes. So yield, you need to identify what are the QTLs that are there, if they are identified or not, if they are mapped across the genome, and then eventually you need that system where it helps, all these help as an attractive source for potential targets for genome editing. And there are several ways you can target. So I'll just go through these fast. So this is the first strategy where gain or loss of functions are utilized. Either you can create frame shift mutations through any of these. We are talking about Cas9 here. So you can use any of these. These are protein based. Zinc finger nucleases and talons are protein based. So you require, for every gene, you require a specific protein module, and it is more time consuming, cumbersome. But in case of Cas9, you just require that 18 to 20 base fragment sequence of the gene, and you require the PAM sequence, the NGG around with it, and then you can utilize it for editing or creating double strand break. So there is a frame shift here, which because of the double strand break and non-homologous end joining repair mechanism, there is a frame shift and you create a mutation here. Similarly, there are amino acid substitutions can generate partial loss of function or gain of function alleles. Then there are screening. There are ways, if you know the protein functional sites, if you know a functional site of a protein, you can screen those key functional sites in the protein by creating tilling guide RNA array, overlapping array, guide RNA array of the whole genome, and then create a library out of it. And then you can utilize base editors or prime editors or Cas9, whichever tool you require, you have to based on that, create a transform the bacteria, and then screen the pooled transformants through the regeneration. The mutant population can be screened to genotyping eventually for beneficial alleles. So here you can see cereals with high amylose people have created. Okay, high amylose content, because amylose is a resistant starch which degrades slowly, and hence sugar spikes are not created. So it's good for diabetic patients. And then oleic acid people have tried to increase. I told you how, because it increases the smoke point, and this offers beneficial benefits for human health. So there are other aspects also. People have created waxy genes where they've tried to stop amylose content instead and create amylopectin, because all the sugar eventually forms either amylose and then amylopectin A. So if you want if your utilization is in the form of amylopectin A, which is important for creating corrugated sheets in the factory or something, so you require more of amylopectin A. So hence you knock out the production of amylose by just one gene. The waxy gene is knocked out, so the entire sucrose goes towards the amylopectin A, and whatever they require for these sheets and for for paper making, for textile corrugating, and for adhesive industries, all this required. So then also, if you create a knockout in a BADH, they have shown a BADH2, betaine aldehyde dehydrogenase aldehyde. So betaine aminobutyride, whatever is that, the GABA is stopped, GABA production is stopped, the entire gamma aminobutyraldehyde goes towards pyrroline, and pyrroline is then eventually the precursor of 2-acetylpyrroline, which gives you the aroma in the basmati rice or something. So if you want to increase the aroma, you will knock out this gene and it will lead to increased aroma. So this is all I'm giving a overview, like people have tried to knock out the vitamin genes said in 2 different places, like Kathy Martin's team at John Hines and knocked out the 7-DR2 gene to increase the vitamin D3, pro-vitamin D3 content in the tomato fruits. So an easy way to provide vitamin D3, that they wanted that, because this vitamin D deficiency is a problem in the real world, because people always sit in the rooms and AC rooms and labs, but people do not go out in the sun. So we need to have the vitamin D naturally absorbable or bioavailable in the body, because if you have tablets, maybe generally vitamin D, how to take it? Do you want to take it in 60K dose, or you want to take it 2000 every day, 2000 units every day? So that is always a bias and a confusion, and the doctors keep changing according to the whatever the Lancet or the New England Biology journal will tell you. So if you eat eggs, if you don't eat eggs, and if you want a natural, vegetarian diet, so these can be a source of. So you can read this paper, there is it is available. Then there is another scientist called Sung-Wei Cho from Seoul University, where he tried to knock out the 7-DR1 gene here. So this is basically the brasinosteroid pathway, where the brasinosteroids and brasinoids and sigmasterols are produced. So he tried to knock out the precursor, this gene that converts the cholesterol into brasinoid and sigmasterols. So these eventually also lead to the higher production of vitamin D, pro-vitamin D3, according to them. So there are duplicate pathways. So if they have targeted 2 different pathways, and they've seen they analyzed this thing, and they found that there is increased amount of whatever they wanted, this pro-vitamin D3. And when you subject it to UVB light, these slices or tomatoes, then they increase the production of vitamin D3, which is the absorbable form. D2, which is found in fungi and mushrooms and all, whatever yeast and all, that is not absorbed by human body. So then this is another way we could create loss and gain of function. Several traits have been used. I'm moving through it because we have less time. So this is targeting, you are replacing the conserved region by whatever mutation you require, that we have done in our lab, that I'll show you the example there. For herbicide resilience or ALS gene, astrolactate gene, I was utilized. So they have replaced the conserved domain with the mutated domain through knockout, and then knocking in utilizing the homology donor repair template with the desired mutation. So there are several, also there are people have created mutations in their in the promoter regions. They've tried to create mutations in the promoter region, swapping the promoters, utilizing high expressing promoters to overexpress the Argos 8 protein, which leads to resilience to stress in the field in maize and all. So then base editing, as I said, there are 2 base editors that have been identified. One is David Liu, the scientist has done that at MIT Broad Institute. But eventually there were certain, drawbacks to it. There were also mutations at the RNA level. So they wanted to screen, refine them. So as of now, they have just not went forward. They haven't gone forward with this strategy, but there is a strategy to, just for base editing, but that has been eventually taken over by, here you can just convert 4 kinds of conversions you can have. C to T and G to A to G, or T to C. This is through your cytosine base editors. And then there through 8 adenine base editors, it is A to G or T to C, or C to T back, and G to A back. So this is basically, the process by which there is a specific, activator domain there. So there is a cytidine base editing domain here, and there is a adenine base editing domain attached to the Nickase Cas9. Nickase is where is the Cas9 where one of the domains is not functional. So there are two domains that create two different double-strand breaks by two different domains, RUVC and HNH. So in this nickase, there is one of them is not active. So they will only create a nick, not a double-strand break on both the strands. So they, depending upon that, they have utilized this strategy. But then came the primer editing technology, where you can have all the 12 transitions and transversions can happen. So you can go through this paper eventually, and they have utilized the Cas9 nickase platform, where the reverse transcriptase along with it is put into the cell, into the and then that reverse transcriptase helps read the inserted domain that has been introduced, that is the edit, that is the template for the reverse transcriptase, and this leads to the formation of the introduced mutation in the cell. So this has a labeled as prime editing, where all kinds of A to T, T to D, T to C, G to C, all transversions and all transitions and transversions can happen. So I'm moving through this because it will be there in the paper. You can go through. Then the second strategy is the genome editing tools to regulate the transcriptional level, at the transcription level. So these are the different ways, and out of these, I would say you should go through these. They are all important strategies. There are introduction of random deletions in the promoter that can, because now the promoter is the main region that controls the expression of the gene. So if you want to overexpress a gene, you have to change the promoter cis elements to those elements that can actually lead to increased expression of these coding sequence. So introduction in random deletions, the promoter can lead to allelic series that eventually have different levels of you see the expression levels. And then you can have targeted disruption of the transcription factor binding sites, because the transcription factor is a protein that goes and binds to the promoter and activates or deactivates it. So that ways that can happen. You can utilize the regions where this transcription factor binds. For example, in the case of blight, okay, or the Xanthomonas, which targets the rice, it basically releases type 3 secretory proteins that are basically transcription factors that go and bind to the promoter regions of specific host genes. And those genes are then labeled as host susceptibility genes. And those promoters of those genes are, taken over by these transcription factors of the secretory protein type 3 from the zoo, the Xanthomonas or rhizae, that eventually leads to the disease called blight or whatever. So blast, sorry, whatever that, there's a blast and a blight. My throat is giving way. So this is there are all these aspects where you can introduce a certain enhancer in the promoter region, okay, or you can introduce a deactivator in the promoter region to increase or decrease the expression of the gene. And here it is not the transgenic way. It is like genome editing is superior than transgenic, because here you're not introducing anything. You are just modifying the native genome of the crop. You're not introducing any antibiotic. You're not introducing any exogenous promoter or gene from other sources. Then comes the epigenome editing, where you can change the DNA methylation domain. So there is a dCas9, which is a dead Cas9, where both the double strand creating domains RUC and HNH are deactivated. So here no cuts are being created, but it acts as a platform to connect with the bind with a DNA methylase or a DNA demethylase domain, which actually can control the expression of the genes eventually. Demethylation will lead to expression of the protein, and methylation will lead to the non-expression of the gene. So that is how you can control by controlling the promoter regions through epigenome editing tools that may activate or repress the transcription in the process. So So these are also being utilized to, live cell imaging you want to study. You can use these. You can, tag them with different like mCherry or different GFP or other, what do you say, fluorescent dyes, and you can just see when they're tagged, you can see where the protein is being gone. So you have gene activation, repression, or genomic labeling, both, all these three you can study. So these are all aspects that have been utilized. So different ways, diverse tools for diverse, modifications that you have to choose and decide amongst the plethora of, tools and in the arsenal of CRISPR. So genome editing tools that can modulate gene expression, there are at the post-transcriptional and translational level also. So there are certain mRNA binding sites, which are there across. Sometimes they are at the 3' end, sometimes they are the 3' UTR region. So eventually you can modify them, and then you can modify the binding of the mRNA. You know how mRNAs work as master switches, so you can modulate the expression. Then there is base editing you can utilize for the changing of bases in the intron or donor site or the acceptor site, and that will lead to interference in the mRNA splicing. So again, the expression will change. Then you can also eliminate upstream ORF or open reading frames that eventually, are controlling or stopping the downstream expression of different genes. Sometimes one ORF is controlling the downstream expression of 4 genes. And if you knock that out, or create a mutation in that, then the downstream expression of several genes can increase. So there is deregulation, overexpression. Then insertional and translation enhancer elements can be utilized to increase the translation. So these are all aspects that there are examples. I'm going over this because you see, this is one of the examples where the translation upregulation was, URF was modified, and the translational regulation of the UORF was modified, and this led to the increase in the lycopene content eventually. So that is what was shown. The mutation here was created, and eventually this hold or the control of this URF on the downstream genes was released, and this led to the increase in the lycopene content in the vitamin C content, the ascorbic content so they'll try to utilize the genes that are eventually affecting the downstream gene expression for ascorbic acid content in the ascorbic acid biosynthetic pathway. So then there are certain things you have to look keep in mind about reducing the off targets. I told you it has to be targeted on target, not off target. So you need to design your guide RNA based on the guide RNA. Your off targets will be reduced. You have to see that there are no matches. There is minimum off target effect if you design the guide RNA properly, utilizing the metrics that have been generated through different mainly animal based research, not much of plant based research, but anyhow it can be utilized to predict the off target sites. And people have also tried to modify the endonucleases PAM site, the NGG, to make it less restrictive. So they have tried to increase the usability. So these are canonical PAM sites. So try to make it less canonical and make it less usable. So there are sometimes NG, you will have certain PAMs that are not identifying NGG but just NG. So that is easier. So there is more targets available. If most of the time the target is based on the NGG. If there is no NGG there, you cannot use that target sequence 1820 basis. So if this is just NG, then that is less resilient, less stringent. So that is also, there are other ways also, CPF. So these are all there. You can go through the papers here that if when the bacteria have the CRISPR, the viruses have developed an anti-CRISPR technology. So there are anti-CRISPR proteins, ACR1, 2, 3, and they have their different roles are to negate the Cas9. So they also can be utilized to control the Cas9 activity window. So I'm not going deep into this. You can read about it. And there are other ways to reduce the off target by utilizing ribonucleoprotein strategies, where you just use the Cas9 protein and you use a kit and you make the in vitro complex on the bench, and then you can shoot it into the plant. So the Cas9 protein will do its work, but it will be eventually degraded. So there is no backbone, vector backbone left. So there is no process of trouble to remove your backbone and all, and then your whatever. DBT requires zero exogenous material in your this thing edited lines, so that can take care of that. So then, so this is, and you can you can read the papers here. This is basically how the RNP is done. And sometimes people have also tried to enhance the homology donor. I told you homology donor repair is very happens at a very low frequency, so you need to boost that and reduce or inhibit the NHEJ so that you have precise modifications, not randomly error prone modifications or mutations due to NHEJ. So sometimes this is also utilizing the FOC1 target. They utilize certain dominant negative domains. They fuse that to your DCAS9 and all the nickase, and then you use that as a platform to enhance homology donor repair rather than your NHEJ pathway. So they've done it in different, not in plants, but they have done it in other animal cells. So here, this is again how you can develop a CRISPR-mediated genome-wide screening strategy where you can eventually create a TILling or guide RNA library, make a vector library in that. You clone all these guide RNA into it, and then you shoot it. And once you see the regenerates and see the phenotyping, as per the phenotyping, you can then choose what to sequence and see what is the mutation happening there. So this is like reverse genetics and then forward genetics के जैसा है. So you can read Siddiq et al. So here this is again, there are several others, so you can go through this. There are ideal strategies also. People are using nanoparticles, and particle bombardment is one strategy, agrobacterium is other strategy, but there are viral DNA delivery also is happening using viral vectors, where there are different platforms. So these are different genes that have been used to regulate. Sometimes genotype is a limitation for transformation, so they have utilized this baby boom and WUSCHEL2 gene mutants already developed in this variety of, maybe this are maize variety or a rice variety, whatever. So they already use these mutants to leverage them for genome editing. So here the genome type limitation is overcome and the tissue culture period is reduced. You can use pollen as target, seeds or target as shoot apical meristem, cotyledon, hypocotyl, whatever you want. Even the embryo you can isolate and use. So the delivery targets can be different. And see, there are different technologies that now I've summarized here: gene knockouts, gene knockout, and knock in, like replacement through homology donor repair. And then there is a base editing. There is fine tuning where you can tune the promoter region by introducing enhancers or modifying the upstream ORFs and so on. So then all these aspects are being covered, and different kinds of traits have been introduced using these technologies. And you can see there are viral resistant chocolates have been developed, then fungal resistant bananas have been developed, even grapes, fungal resistant grapes, resistant to powdery mildew. This is fusarium, resistant to fusarium banana have been developed because Cavendish banana was giving in against this fusarium, but now they have developed resilience to that through genome editing. Corn and rice producing more grains under drought conditions or heat conditions or whatever. So then coffee that is decaffeinated. So there are some people who do not want to have caffeine, so they are allergic to caffeine or whatever. So you can have decaffeinated coffee. Wheat has a lot of gluten and gliadins, and that are also creating a disease, celiac disease. So you can overcome that through eating these. The Spanish scientists have developed, I think they have knocked out, more than 100 genes, and they're trying to test them now. So mushrooms that are anti-browning mushrooms, they produce tomatoes with different tastes and critical flavors. They have been introduced. So this is all basically, utilize, we need to create our own, boost our own food industry and the amount of nutritional enhancement that requires for our country to become Atmanirbhar. So this is what one of the tools to focus on this way of getting Atmanirbhar or becoming Atmanirbhar. So I just wanted to just showcase few of my products, and then I will wind up for the questions. So these are the different site-directed nucleases that are allowed by the DBT, Department of Biotechnology. This one is not yet allowed because it's more than 220 base pairs. Here, this is knockout and knock-in. There's a few base pair changes, less than 20 base pairs. And here, this is knockout. There are no knock-ins or no. So these are all insertions and deletions that are endogenously happening. So that comes as SDN1. So we have developed some of these SDN1 and SDN2 in different crops. So we have developed a kind of a direct seeded rice that we are trying to create because of see whatever the reasons, the government of India, loss of water, too much water is being taken up in the puddled rice, right? The transplanted rice. So transplanted puddled rice is a major contributor to global warming and water shortages, low groundwater levels in various cities. So this is the best alternative to that. So it would reduce the water usage and reduce the methane emissions, but it would all increase weed rivalry and micronutrient deficiency. So we've targeted these two traits, and we've created herbicide resilient crops, and we have tried to increase the iron and zinc content in cereals. So I'll just go just show you that this is the main pathway that we have targeted. This is the shikimic acid pathway, which is basically creating the essential aromatic amino acids, phenylalanine, tyrosine, and tryptophan. And these amino acids basically are important because without their presence, the plant will die. If the weed dies, so the strategy to kill the weed is that you spray glyphosate, one of the inhibitors of this enzyme, the sixth enzyme of this pathway called the EPSPS synthase. So EPSPS or EPSP synthase is the sixth enzyme, which natural substrates are shikimate 3-phosphate and phosphoenol pyruvate. But as glyphosate mimics, it is an analog of PEP. It is similar structured analog of PEP. It's a competitive inhibitor of this natural second substrate of this enzyme. It goes and binds to this place instead of this. So the enzyme activity reduces. Hence, these aromatic amino acids are not produced, which eventually leads to the death of the weed. But our main crop also has the same pathway in the chloroplast. So they are also affected with this inhibitor, glyphosate. Hence, to give resilience to our crop, while we are spraying the glyphosate in the field, our crop should not die, or it should not reduce its efficacy of production higher yield. We have to introduce this pathway where we have created mutations, 3 mutations in this active site binding domain. So that has led to, this is how we introduce, this is a natural mutation, the conserved region. So we created 2 guide RNAs flanking this conserved region, and we created a knockout. And then we gave a homology donor template, which had the desired mutations in the center with the left homology arm and the right homology arm, which is totally, same as the gene here. So when this thing goes in, the endogenous machinery recognizes that this is the part of the same gene from the left arm and the right arm, and it throws into this gap this piece of DNA, which has just 3 mutations, right? So that is how SDN2 is created. So this is precise modification of the EPSPS gene, that the mutant native gene itself is mutated, not any exogenous overexpression of any exogenous gene from any bacteria or whatever, the CP4 bacteria that people are being using for soybean, GMO soybean transgenics. So that CP4 bacteria, Agrobacterium gene, which is naturally resilient to glyphosate, that was overexpressed on these genes. And then they called it Roundup Ready soybean, Roundup Ready, whatever. So here we are not introducing any CP4 gene. We are just editing the native gene in the crop. So this is the sequence. See, these are the 3 regions that were to be mutated. So we mutated these target genes, the target regions, replaced it. So the G became A, the T became I, and the P became, the proline became serine. So these are the target sequences that I showed you. We created 2 guide RNAs flanking this region, and we removed this part. And then through the homology donor template, with the desired mutations, we introduced the mutation. So this is the wild type. And now you see that DGA became GCA, and the ACT became ATT, and the CCA became AGC. So these are all different mutants, edited lines. And here you see the Cas9 gene also segregation happened in the T2 generation, and we got these lines minus the Cas9. So the yield was enhanced by 20 to 22%. So eventually you see when the mutation we introduced in this site, what happened was the glyphosate became insensitive to this crop, but the binding of PP to its enzyme enhanced. Binding affinity of phosphoenol pyruvate enhanced, and this led to increased production of phenylalanine, tyrosine, and tryptophan. So this led to an increase in the aromatic amino acid profile and growth of the crop. So here you can see the initial substrate, that is shikimate, was better utilized in the edited lines than the control. And this treated control eventually dies because after 20 days it cannot survive because the essential amino acids are not produced, and hence the plant generally dies. If it is too much sprayed, the tip starts burning and it dies. So, but eventually these lines survived. Okay, and then you see the essential amino acids also increased. Aromatic amino acids increased here in comparison to the wild type and the treated control. So this is a kind of a protein. So this is how we show that we aligned these different conserved regions. You see this is the conserved region, which was aligned for different crops. Then you can see what mutations. The wild type is the mutant, and we introduced these 3 mutations here. That led to the change in the cavity size. You see there's a cavity size here, and here it changed in the mutant. So in this change led to non-binding or reduced binding of glyphosate here, but increased binding of PP. So that is why the amino acid profile increased. So this is this is just a comparison between the. So we sprayed the glyphosate here. You see the edited lines before spraying glyphosate, and these edited line mutants, events 2, 6, and 9 after spraying glyphosate. So you can see they could tolerate up to 1.5 to 3 ml of glyphosate. Okay, and this at this concentration, the wild type and the weeds died. So this is the control plant before spraying glyphosate, and after spraying glyphosate, the control plants all die at 1.5 ml Roundup and 3 ml Round. And this is the so there, and then we developed high iron zinc lines in the rice, which is actually accumulated in the central endosperm portion. You see, generally this portion is starchy, and the outer this thing is removed through milling and polishing. So in the wild type, you see how it is, and in the edited lines, how it is enhanced. So we checked the agronomic parameters and all, and we have these Cas9 free lines, and we checked. Eventually, I'm just forwarding how it increases. The iron and zinc is going inside, and the nitrate is going in the biomass. We checked for the nitrogen content also in the edited shoots. So this is like biofortification, and then cobalt and cadmium was reduced in the shoot. It did not go in the edited shoots, but it got stored in the edited roots. So it was like a bioremediation effect. And then we have different other crops also we have developed. So we have increased the herbicide resilience in pigeon pea, multiple herbicide resilience in pigeon pea lines. Then glyphosate, we enhance, herbicide resilience to glyphosate in corn. For rice also we developed, which I have shown. Then we are doing it in soybean also now. Then we reduced profilin, which is an allergenic protein in the tomatoes. 20% of the people population is allergenic to profilin, and they cannot eat tomatoes. Maybe we do not know much of them, but there are many around in the world. So this is how cotton yield we have enhanced, the tensility we have enhanced. Okay, then comparative flowering, early flowering we have introduced in Vigna rice bean, which is a very rich, nutritiously rich legume, but not eaten mostly in the Northeast. And then we try to edit the saffron to produce more flowers per corn, right? And then as you all know, the rice and all. So this is the corn pictures that we have. The edited lines after spraying, they survive 3 ml per liter. And most of the, yes. So most of the plants survived, See, yeah, they recovered after 45 days of glyphosate resilience. After spraying glyphosate, they recovered, but the control plants died. So most of it you can see in the figure here. You have published this also. This is a small greenhouse that we had in the plots showing these edited lines. And so this is the same way we introduce these mutations in maize. We grew the lines and we have a patent on this. So this is the mutation that was introduced here. These are T2 lines. The agronomic parameters were better in the edited lines as compared to the treated control and the control lines. The control lines are without spray and the treated control is after spraying. The same line after spraying. And these are the edited lines after spraying. So they are working better than the treated controls. And then we also saw that there is the enhancement in the aromatic amino acid profiles in these corn lines again through LCMS we checked. So here you can see after 3 ml of Roundup sprayed, in the plots, we could see the edited lines could recover within 45 days, but the control lines could not. So we have a project with for developing multiple herbicide resilience to different other herbicides in corn with the company. And we had again also we have also developed these multiple herbicide resilient lines that are resilient to inhibitors of EPSPS and ALS and Pigeon Pea, that is the Arhar dal, the some used for the sambar. On the same strategy, we've isolated 4 mutations. So this is G to 8, T2I, A2V, and P2S in here. And here we have a P2S and a W2L, which gives resilience to multiple herbicides. So now why it is required? Because continuous spray of the same herbicide again and again, leads to the development of herbicide resilient weeds. So we need to change the herbicide spray in every season, so that the resilient weeds do not come up. So in US and other countries now, glyphosate is not effective because the weeds are resilient to glyphosate. So now glyphosate is not being used. So they look for other herbicides that are, for corn and for soybean and for canola that are resilient. So they are all looking for ALS and other different 2,4-D, dicamba and other herbicides. So now we are looking for different mutations in those target genes. We are doing it in this project. So this is the multiple herbicide resilient Pigeon Pea lines. Okay. This is sequencing details, how we introduce the 4 mutations and these 2 mutations in the ALS gene and the 4 in the EPSBs gene. Here, this is in the jam bottle. You could see the multiple herbicide resilience in the wild type and the edited line. The first row is the wild type WT and the rest of are the herbicide edited lines. So there are different herbicides being sprayed here. They are all inhibitors of this one is the inhibitor of EPSPS enzyme, and other 4 or 5 are inhibitors of the astolactate synthase enzyme. So you can use imazepine, imazethhyper, chlorsulfuron, bisparax sodium, chloransulam, methyl, flucarbazole sodium. So you can use all these to spray. So the farmer has a choice now. So we need to just on the packet, we have to label that for this season, you can use this and this or whatever. That is up to us how to label the packet. So these are the T2 lines, the T2 generation. And this is now the lines that are now, we've identified, growed them in the field. And we've seen after spraying, the wild type is dying, here. So these are the three mutant EPSPS lines. Okay, here. These are the 3 the SH is a single single gene mutants where only the EPSPS gene is mutated. So we are just spraying the EPSPS inhibitor here, but this is the single gene mutants and these are the double gene mutants. We have not yet sprayed the ALS herbicides. So as of now, when you see up to 4.5 ml of glyphosate, Roundup, they can tolerate. So after 30 days, they recover also, but the wild type totally dies. Okay, this is the unsprayed ones without spray. This is the sprayed ones, wild type. And these are the edited lines that are sprayed with different concentrations of glyphosate. So they have recovered after 30 days. So this is for soybean. Again, we have similarly, we're doing it for EPSPS and ALS genes. These are the T0 and the T1 lines for ALS mutants. And so simultaneously, we are creating, because there are 3 isoforms of ALS and soybean. So we are trying to, do that now, but we are in the process. But we've got the EPSPS lines, resilient lines, and this is the T1 and the T1 lines for the ALS mutants. So again, these are the Vigna umbellata. I told you rice bean mutants for early flowering, because it's a late flowering, and in the hilly regions there is late flowering and there is no flowering sometimes. So when it comes to the plains, we need to, or in the hilly region, we need to make it early flowering. So we're trying to induce early flowering and a medium size height, because it's too it becomes spiral and it just goes keeps on growing on the wall. So we need to reduce the height. So that is what we are targeting. And the palatability issue is there. It's not palatable. There are certain aspects, like there is phytic acid, and then there is hemathrine, and there is trypsin. So there are all these antitrypsin. So they are all, creating unpalatability issues. So we are trying to reduce that also through gene editing. This is the cotton one, where the student has himself developed these edited lines. You see the control and you see the edited lines. They have longer fiber length, and the tensility is also increased. So they are stronger. And so the fiber quality and quality both have increased. This is the lycopene enhanced profilin reduced tomatoes through genome editing. And this is a team, my team. And you see Sonia from Bangladesh, Mamta, Arul, he's from Tamil Nadu, Khalid from Egypt, Josna, Rachna, Murgesh. So Rachna and Josna worked on the double herbicide resilient pigeon pea. Arul developed the cotton and the maize lines, and now he is developing other different cotton lines also. So this is rice thing also with Khalid. He's developed the high iron and zinc rice. Sonia developed the glyphosate resilient rice line. Mamta developed certain chlorophyll mutants that showed increased enhancement in their photosynthesis. Rashmi developed the tomato high lycopene reduced profilin tomato lines. And now these are the new students. Pooja is working on rice bean. Shubhra is on mustard. Isha developed the soybean lines. Sugyan is working towards reduction of the amylopectin A, so that the amylose content is high and it doesn't create spikes, and it's good for diabetic patients. And these are the international fellows. So thank you for your patience. And it's all I give all over to the Pragati madam, and the students can ask any question if they are still not sleeping. I'm sorry for a long.
No, ma'am, they are still awake and they are listening to you very carefully.
Thank you. They're very kind and they are patiently, listening. Very kind.
Yeah.
Thank you. So any questions, I'll be happy to answer.
Yeah, the participants can unmute yourself. You can ask questions directly to ma'am, or you can drop your questions in the chat box too. And just be quick.
Hello, good evening, ma'am.
Yes, 1 second. I just opened my video. Yes, sir. Good evening.
Good evening, ma'am. Myself, Ajay Sharma.
Yes,
I
sir.
have a query, ma'am. Biofortification aim to taken hidden hunger. Basically, it's one of the biofortifications aim. How can CRISPR improve the nutritional quality without negatively affecting the crop yield?
So that is what I said. You need to utilize these new tools based on artificial intelligence, generalized intelligence, and machine learning. So, and these tools help you to develop the algorithms that can predict which genes you should identify or utilize to, increase the biofortification. And there is always a trade-off between the nutritional aspect and the growth performance. So then you can, see balance the trade-off and utilize these tools to predict. And then once you have that, you can do GWS or genome-wide association mapping also. Check whether there are other aspects like any molecular markers that are there that are identified as negative regulators already. Those can be utilized, and then they can be through genome editing can be done, and then screen phenotyping can be done, and then eventually those that are actually working well in the simulated conditions, because we cannot go directly to the field. So in the simulated conditions, if they are working well, then they can be further given to the breeder to see it in the field evaluation phase.
Okay.
Yeah.
Thank you. Thank you, ma'am.
Any more questions?
Yes, ma'am. There is one question in the chat box. Ruchika is asking, very insightful lecture, ma'am. I have a question. How GM crops are different from CRISPR-Cas9 technology, or are they similar?
So a very good question. I finally found that there are several. The lecture doesn't cover most of this thing, but I did explain that when you are talking about GMOs, they are basically based on introduction of exogenous genes. So there are exogenous genes that are utilized. For example, if you talk about golden rice. So what was golden rice previously? It was a conglomeration of 3 genes that were taken. 2 of them were taken from a flower, daffodil, and 1 of them was from a bacteria. Right. So they were introduced, they were also developed as a overexpression of these genes were because the vitamin A biosynthetic pathway has a bottleneck, and these 3 genes were there in the upper part of the pathway, the phytoin synthase and 2 more genes that were there. And these were overexpressed using a highly overexpressing promoter. For example, 35S promoter, maybe, I don't know, the 35S promoter, which is from the viral promoter, which is. So this eventually creates a transgenic approach. Why? Because you are creating a construct, you're utilizing an exogenous promoter, an exogenous gene from another crop or another soil organism, and then you're introducing it into the crop, rice crop, right? So that is a GMO approach. But when you are editing the native gene of the rice, as I showed you, that in this case, in the EPSPS case, I showed you, we are editing the native gene. We are not introducing any mutant. I told you I have not introduced any CP4, gene overexpressed in this, because the strategy that the company used, by Monsanto was overexpression of CP4 agrobacterial gene, which is naturally resistant to glyphosate. That was overexpressed in soybean first time in 1996, and they developed those GMOs. But our case was not. So our resilience to glyphosate is maybe less, but it is resilient enough to protect them from spraying glyphosate. So the native gene is modified. I showed you the slide. I'll show it to you again if you think you do not remember. I'll show you that slide. I knock out and knock in. Where is this? Where is it? Yeah, this one. You see, so this one, if you see this, so I showed you here, this is the conserved region. This is the native EPSPS. It's not any exogenous gene that we are introducing. Rice native EPSPS gene. We identified the conserved region. We created 2 guide RNAs, removed this piece, then introduced these 3 mutations in that place of GTA. We introduced AIS by utilizing this homology linear template, and that eventually had the flanking regions, the left arm and the right arm, that were totally similar, same to the native gene, just to help just to nudge the pathway towards homology donor repair, because I told you most of it is done, the repair, endogenous repair is done through non-homologous end joining. Hence, to nudge it towards the low efficiency homology donor repair, we tried to create this template, gave this template at the time when this was knocked out, and then through site-specific homologous recombination, it was introduced inside the native gene, and then you call it a mutant gene. You understood this? Did you get my point, or should I have to speak more? Hello, Ruchika. I hope it satiates your answer. So they are better than GMOs because we are not introducing any antibiotic gene, any selection marker. We are not introducing any exogenous genes from bacteria or any other crop. Only the native gene, the host plant gene only we are editing. The background itself we are editing. Okay. So in this
Yes,
way,
ma'am.
it is
She's
as
saying thank you.
as the wild plant, but with a different trait, without any exogenous material, without any marker, without any extraneous material, right? Yeah.
Yes, ma'am. She's replying. Thank you so much, ma'am, for the explanation. There is one student, ma'am, Yugdeep. He's asking, ma'am, what path did you take from your BSc to your current position, and what advice will you like to give someone who wants to pursue MSc in plant breeding, foreign or Indian?
So if you really want to jump into this arena, which is very tough, so you need to either go for a specific whatever aspect you want to work on. You need to identify if it's an agricultural problem that you want to seek an answer to, then you need to go to the agricultural industry, agricultural university. For example, ICR is there, and so there are MSc programs are there, integrated MSc programs are there. You can work there with the scientists, and then you can create a pathway towards genome editing and all. Then there are research institutes like ICGB is there, the NIPGR is there, IBRICS institutes, RCB, THST, they're all there. So you can go for, these are all good institutes within the country, and ours is an international institute. So you can have a PhD through, after your MSc, you can have your PhD with us. We have a MOU with JNU, so they give the degree, so the degree is through JNU. So eventually, if you want to go out, then you have to do some clear some exams like IELTS, English exams, or maybe some exams you have to give. For every university, there's a different exam. So if you want to do, but I would suggest if you want to go for a PhD outside, you need to have some kind of a training somewhere. After your MSc, you can do some dissertation work, or as a project associate, you can stay with some national labs, learn the techniques, and then you can apply for a PhD outside. So as of now, the situation, you see the World 3 like situation everywhere. So I do not suggest in the next 2 years, but eventually you're still young, so maybe after 2 years you may go out. So you can just do your MSc and MS, or your training with some labs as a project associate, and then learn the techniques, and then you can apply after 2 years. 1 or 2 years basic research you need to do, and applied also wherever. So there are numerous ways you can. So there is no single path, right? So you have to think. Most of the people want to go out, but now the times are like their fellowships are not very abundant. So people are not giving you fellowships, but they are asking you to bring your own money and take care of the staying expenses. So if you have that kind of money, you can go choose the good universities where they offer you, genome editing platforms, like MIT is there. So you can choose such. The Scripps Institute is there. There's so many. New Jersey, there are so many. New York has several good. Then this California State University is good. There's several of them. Even in you can personally take my email ID. I can help you out with that. So, thank you.
Ma'am, students are requesting for the study material, the presentation you have used, if you could provide them.
I will send it to you, then you can distribute. Okay.
Okay, thank you so much, ma'am.
Review papers. I told them if you can just go through the review papers, I had given them, but I can send you if you want the PPT, I can send you the PPT or the research papers, whatever I made from that, I can also send. Both.
Okay,
Yeah.
okay, thank you so much, ma'am. I think the questions are over for now.
Thank you, Pragati. Thank you, Pragati. Thank you for this opportunity. Thank you for the patient, clear audience. Okay. Yeah. Thank you.
Thank you so much, ma'am. Revangini, please conclude the session.
Thank you, everyone.
Okay, ma'am. So, on the behalf of entire organizing team and all the participants, I express my heartfelt gratitude to our esteemed speaker, Dr. Tanushri Kaul ma'am, for such an insightful, inspiring, and knowledge-enriched session on genome editing like CRISPR technology. Ma'am, despite not feeling well, you still took out your valuable time to join us today and share your immense knowledge and experience with all of us. We truly appreciate your decision, kindness, and commitment towards the guiding young man. Your scientific vision, motivational words, and remarkable contributions have inspired every participant to think beyond boundaries in the field of agriculture and biotechnology. I would also like to sincerely thank all the participants for their active participation throughout this session. We hope today's interaction become a source of inspiration, learning, and motivation for your future journey. Thank you so much, ma'am.
Thank you so much. Thank you so much, the August gathering, for your clear audience and for your patience. And my email ID is there at the website, tanushri@icgb.res.in. The ICGB website has my email ID. You can put your questions if you have any more. Somebody who could not answer, ask. I'm putting it in the chat. You can see it. Okay. Thank you so much, Pragati.
Thank you so much, ma'am, for your cooperation. Thank you, ma'am.
Thank you. See you then. So I take leave.